Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy, and I'm in the Communications Department at BTI. And for this month's Breaking Ground, we welcome Dr. Andrew Nelson, an RNA biologist and genomicist here at BTI, whose main research focuses on how plants respond to stress. And today we'll be talking about Andrew's research projects with a bunch of different plants. And hi, Andrew.
Hi, how's it going?
Pretty good. How you doing over there?
Good.
Before we get going, a few housekeeping announcements, which those of you who've joined us before are probably familiar with. During the session, all participants will be muted. So Andrew and I will have about a half-hour discussion, followed by half an hour or so Q&A, and you can ask questions in the chat. And then at the end, we'll do a Q&A session at the end of the conversation. And I've enabled a live transcript function, so you should be seeing closed captions. And if you'd like to turn that off, you can do it with a live transcript button down at the bottom of the screen. And yeah, thank you all for joining us today. It looks like we got a good crowd out there. All right, so I guess to kick things off, Andrew, could you tell us a little bit about yourselves and your research?
Sure. Yeah. So I'll share my screen briefly. Just to give a little bit of background about myself, I've kind of lived all over the place. There's an expression called in America, army brats, and typically you expect army brats to have lived all over the world. There's a similar expression called oil brats, and so my father was an engineer for an oil company, and I also lived all over the world, wherever you kind of expect oil to be found. But for my undergraduate experience, I went to a small state school, Southwestern Oklahoma State University, in the middle of nowhere, Oklahoma, with the intent of going to veterinary school at Oklahoma State. So this was a way for me to kind of a bridge to Oklahoma State. And when I was there, I realized that, veterinary school didn't really appeal to me for a number of reasons. And instead, working or performing research with three completely different scientists with completely different backgrounds really, it made me really interested in science as a fundamental thing that I wanted to do in the future. So I did field work, I did biochemistry, I did genetics, type of research, and it was a really just a good experience. And so I went from there. I went to Texas A&M in the lab of Dr. Dorothy Shippen to do my PhD. Working, that's really where I started doing my research in RNA biology. Worked with a bunch of really, really amazing people. Spent a few years there, and then went to do my postdoc at the University of Arizona with Dr. Mark Beilstein and Dr. Eric Lyons, two very fabulous co-mentors. There, I continued to do RNA biology work, but I also, with Eric, I really started to appreciate and understand the genomics aspect of things, comparative genomics, like figuring out what's going on in an organism by looking and comparing between genomes. And then I was part of the cluster hire here at Boyce Thompson. Gosh, it's been, I think, a couple of years ago now since I've arrived, almost 2 years. And I was hired alongside two very fabulous people, Magda Jolkowska and Ola Skirich. Again, like we have a very nicely complementing skill sets. And yeah, we've been doing a lot of fun research since. So in terms of my research,
well, can I be quick?
Yeah, and please interrupt. Yes.
I'm curious how you went from interested in doing veterinary stuff, which is animals, obviously,
yeah.
and switching to plants. Did that happen in your PhD? Did it happen before your PhD?
It happened. So I spent if you want to go to vet school, you have to do a lot of volunteering or working with vets, just like if you want to go to med school or even if you want to go to graduate school, you kind of get a taste for what that field is like. And I worked with large animals. I worked with small animals, or I volunteered at clinics that worked with these two different types of animals. And large animals, of course, in Oklahoma, it's mostly cows, mostly, doing a lot of like high throughput surgery on those cows, shall we say. Not the most not the most fun I've ever had. And then the small animals, which of course I love. I love cats and dogs. It just doesn't, being a veterinarian doesn't pay very much, surprisingly. And I realized that, and this was discussion with a veterinarian that I helped with, that he had been in his practice for, I don't know, 10, 15 years, and he just finished paying off his student loans. And he was just about to start paying on his loans for his practice itself. And I thought to myself, well, I don't come from a family with great means. This doesn't sound like a prudent financial decision. And it just so happens that at the same time, I was doing research with these three scientists, and I was getting paid to do this research, and I was getting paid to do something that I thought was really cool. So it just seemed like a no-brainer. You get paid to, do something that you love, which I guess is what most people are looking for.
And they're all plant scientists, those three?
Actually, interestingly enough, none of them were plant scientists. So one worked with a slime mold, Dictyostelium. The other one the entomologist worked with mayflies. And then the biochemist worked with essentially E. coli purified protein. So the reason that I got into plants was another kind of, just one of those random things that you come across. I actually, when I was in my genetics lab, I read this little insert that came with the plasmid prep kits. So these kits that we use are standard molecular biology kits. And this insert was kind of a promotion for research being done in humans on this potential to extend lifespan. So the idea was if we add this this protein back into human cells, we can make them immortal, and we can cure aging. And this was research being done at UT Southwestern, University of Texas Southwestern, by two amazing scientists who had started a company to get going on this research. And I was like, holy cow, this is awesome. This is the research I want to work on. And it just so happened that at Texas A&M, there was a woman, Dorothy, who was working on the same problem, the same topic, but she was just doing it in plants. It's a perfect model system for the questions she wanted to ask. And so that was like roughly 2006. Well, 2006 when I graduated undergrad, and I went to grad school. And as you know, we haven't cured aging yet. And it just so happens that when you take this protein and you overexpress it in mammalian cells, it's actually a hallmark for cancer. So it's just it was an important lesson for me that you really need to understand the system and understand the implications of what you're doing, and that transfers across plants, animals, you name it. So it was a very non-direct route into graduate school for me.
Yeah, that's interesting. It's always interesting to hear the paths people take. There's always a different story. It's always interesting. Okay, so I guess back to your research, you mentioned RNA.
Yeah. So when I was a graduate student, and then when I was a postdoc, we were working on this protein and RNA complex that's supposed to maintain chromosomes. And of course, the RNA component of this complex is an RNA that doesn't produce protein. So it's one of these long non-coding RNAs, is what we call it. And this long non-coding RNA, it was really difficult to find. You can't just with most protein coding sequences, you can actually, if you have the sequence, the amino acid or the nucleotide sequence of that protein, you can usually find it in other close relatives. But with this particular RNA, and with most long encoding RNAs, we can't do that. So they change quite rapidly. And so I was trying to find this RNA in other close relatives of the plant that I was originally working on, Arabidopsis. And I wasn't really able to find it using biochemical, experiments. So I turned to genomics, and I worked with Eric to figure out and see if I could find where that RNA was. And in the process, I started appreciating the fact that there are a lot of these RNAs. There are a lot of these long non-coding RNAs that are found all over the place. So
it's like, I guess if I could just interrupt for a second, I guess when I usually think of RNA, it's usually, you have the DNA of a gene, and then it gets translated into RNA and then transcribed into a protein, right?
almost. It's
backwards?
You got them backwards, and I laughed because I did that on my very first genetics test. I got a C on that genetics test because I got transcription and translation reversed.
I get them backwards all the time. But it's usually like the intermediary between the gene and the protein, and the protein goes and does stuff. But
absolutely. So that is
RNAs, right?
Yeah. So let me share a slide again, and we can unpack that. What we call actually the central dogma of molecular biology, which is that DNA that we all have, is within that DNA are genes. There are little regions within the genome that encode for or transcribed into an RNA. And typically, when we think of RNA, we think of those RNAs that get turned into a protein. So we go from DNA to RNA to protein. And most of us, when we are performing our research, we're actually thinking mostly in terms of the protein, the outputs, because years and years of research has shown that proteins are typically what's associated with the phenotype or the disease or the network of regulation that we're interested in. But it also, I think, probably late 90s, early 2000s, we realized that there are a lot of these small non-coding RNAs, and these non-coding RNAs actually can help regulate the kind of production of the proteins that we may be looking for. And they may also kind of turn off and turn on sections of the genome. So there's a lot of regulation going on by these small non-coding RNAs. But then more recently, we've realized that there are a lot of these long non-coding RNAs that produce no protein, but they still can help regulate either protein production, or they can be involved in kind of helping the proteins do the job that they're supposed to do. So I'm interested in this class of RNA, which I've kind of specifically drawn without this kind of green start and this red stop. This indicates protein coding nature. These RNAs, they don't encode for any protein, but they still happen to do a number of really interesting jobs, particularly in plants. So flowering in the mustard family is controlled in part by a long non-coding RNA. And there are a number of other stories of long non-coding RNAs that I could go into. But my lab in general is kind of interested in these, understanding how RNA in general, and link RNAs specifically, help to mediate stress or responses to environmental stress. So how the plants perceive and cope with droughts or heat or cold. These are all topics of interest to the lab. And so we kind of look at 4 main different things, kind of broad questions, I should say. How non-coding RNAs can mediate these responses, how changes to the RNA structure and modifications to that RNA can influence what the RNA is doing.
So these RNAs can actually be modified themselves?
Yeah, exactly. And we'll talk about that next. But yeah, so RNA modifications, just general changes in RNA abundance and stability. So, we typically use that as a proxy for what's happening at the protein level. And then, of course, RNA compartmentalization. What's happening within the cell? Where is it going? Who's it interacting with within the cell? So these are all things that I'm generally interested in. And as you can tell, it's kind of a long list. I'm interested in all things RNA.
Right. So about some of these modifications, could you talk a bit about those and how those work?
Yeah, so it's kind of interesting. We've known well, actually, when I went through graduate school, the whole idea that your genome, your DNA can be modified by these what we now call epigenetic marks. But the idea that the DNA could be modified was something that was just coming into swing in the early 2000s, and it was this extra layer of complexity that could regulate when a gene was expressed, when it was doing, when it was turned on or off. And it's just these really small chemical marks, these like methyl groups. It's just a carbon and a couple of hydrogens, or 3 hydrogens typically, that are added to the DNA that helps to regulate, whether it's turned on or off. It just so happens that RNA goes through a very similar process. So RNA can be methylated at different sites, and these modifications can help influence what the RNA is doing. So I'm going to show another slide here. It's a little bit busy, but I'm going to try and walk through just a couple of things because I think it really highlights the complexity of RNA modifications. Okay, so we've actually known since probably the 1970s through some really careful and biochemical analyses that tRNAs, transfer RNAs and ribosomal RNAs are highly modified. So what I'm showing here is a classic structure of a tRNA, and all of these little dots are meant to represent modifications that happen almost every single time on a tRNA as it's being produced. And the different colors are meant to indicate that it's different types of modifications that are occurring. So tRNAs, ribosomal RNAs, they constitute probably 97% of all the RNA that's present in a cell.
Oh, wow.
And these things are modified like crazy. And so it wasn't until probably, the last 5 to 8 years that we realized that mRNAs and long non-coding RNAs were also modified. And of course, with tRNAs and ribosomal RNAs, we know that those modifications influence structure. And some of those modifications influence the function of those RNAs. But with mRNAs and link RNAs, we didn't really understand what those modifications were doing. But we did start to, I say we here, this isn't my work, this is the work of much smarter people than me. What we did realize is that these modifications tended to be placed on the RNA in specific regions. So for instance, this modification here, M6A, so this is a methyl group being added to the adenosine nucleotides within the RNA. And those typically get deposited towards the end, the 3' ends of the RNA. Whereas this other modification typically gets deposited within the middle of the RNA. And the location of these modifications helps to essentially has structural and functional implications. That's mainly work from humans. So in plants, we know a little bit about these modifications, mainly in the model plant Arabidopsis, but we don't know much beyond that. However, just I wanted to put in this work from Guifeng Jia's lab in China on essentially altering this M6A modification, altering the ability of that modification to be present in rice here and in potatoes. And what you can easily see here is that when you artificially remove this modification, you get much larger rice grains and you get much larger potatoes. We don't know why this.
Yeah, I was going to ask, why do you suppose that is? It seems like, you know, if it exists, there must be some evolutionary reason for it to
Yes.
making them smaller. Seems counterintuitive.
Yeah, so we don't know why this is specifically in these plants yet, but the way that this group did this experiment was really interesting. They took this gene from humans, which has been linked to obesity. so so they take this gene, and they actually place it, they overexpress it in these two plants, and you actually see some increase, and obviously it's very naive correlation here, but this enzyme is clearly regulating something in these plants. Now we obviously want to know what it's doing. Maybe an easier, for me, an easier explanation is an example from Arabidopsis plants, where we found, well, and some collaborators, Brian Gregory at University of Pennsylvania, found that genes that are associated with a stress response. So say your plants are heat stressed. The genes that help cope with that heat stress are typically, their RNA is going to be modified in the 3' UTR by this M6A mark that I'm showing here. And this mark, what it actually does is it helps these RNAs be translated faster. So you get more of these proteins that deal with the stress response and help the plants cope. And then this other mark, this M3C mark, actually gets deposited on the RNAs, most of the RNAs that don't deal with the stress response, and it actually causes them to be degraded. So there's I don't know what the connection is with potato and rice, but there's some clear gains to be made here. We just need to understand what's going on.
That's interesting. So when they're stressed, they're spending a lot of energy on dealing with whatever that stress is, and if they're not stressed, they can put that energy into the grains or the tubers or whatever.
Yeah. Yeah.
That's
yeah. So plants do, I mean, obviously plants can't move away from the stress. So they have a lot of really clever ways for trying to cope with stress. You know, they can sequester RNAs or proteins into granules within the cell to kind of prevent them from, like, you know, kind of put them away for safe storage until they need them later. But they can also kind of very quickly target RNAs for degradation or for translation through these modifications.
Okay. So could you tell us a little bit about one of your projects? You mentioned
yeah,
Kalska earlier. Are you doing?
Yeah, so let's talk about let's show some pictures. So I talked about long non-coding RNAs, and I talked about, you know, well, I mentioned one specific function or a role for a long non-coding RNA in plants, and there's probably 10 or 15 long non-coding RNAs where we have really good evidence for what they're doing, but we don't have a lot of I say functional data for long non-coding RNAs in really any plant. So what I'm doing with Magda here is I'm trying to screen through, and this is the part of the dissertation or thesis project of Kaylin Rayleigh here, a now second-year graduate student in the lab. And what we're doing is we're using these really nice phenotyping rigs, which is just a Raspberry Pi computer attached to a couple of cameras, and it takes pictures every 30 minutes, and it monitors the growth of plants that are sitting underneath it. So basically you can see here this little wooden contraption sitting up over a flat with a bunch of plants growing in it. So you see the cameras, really small cameras up here. They're actually taking a picture of about half the flat of plants. So we have two cameras. And what you see here is it's actually, you know, taking a picture over time, taking pictures over time, and we can see how these plants are growing, and we can monitor differences in growth rates between plants grown on the same flat. And what we're doing is we're using this to as a means of screening through mutants of long non-coding RNAs in Arabidopsis. So we identified a whole bunch of long non-coding RNAs, and we wanted to understand what they might be doing, in the cell. We have some guesses, some hypotheses, but we don't know for certain. So we're just using this as a really fast way to screen through and find the most promising candidates. So this is an ongoing project. And Kaylin, I think she's on her fourth kind of grow outs, and I think we've identified some promising candidates that have germination defects. But I don't want to put the cart before the horse.
Right. So how do you generate the mutants? Do you mess with a genome? Do you find like where the long non-coding RNA is in the genome and then like knock a piece out of it or something? Or
yeah, so great question. So we're working with probably one of the most densely studied plants in human history. So Arabidopsis has a whole bunch of mutants that have already been generated and are maintained by a couple of stock centers around the world. And the nice thing about Arabidopsis is you see where your link RNA is in the genome. You check to see if there's a mutant that's already been cataloged and already been put in the stock center that overlaps with that link RNA. Now, if we didn't have any mutants that overlapped with our link RNAs of interest, then of course we would have to do kind of targeted editing approaches, what's typically referred to as CRISPR. And that is absolutely something that we have planned, particularly to confirm any of the mutants that we identified to have an interesting phenotype.
Okay, makes sense. And so how do these modifications of link RNA impact plants in the field? How do you link the changes in the field to changes at the cellular
yeah, that's
level?
Yeah, that is a hard word. Yeah, it's and that's a tough question. I mean, it's a really good question. And typically, how you do it is you have to have lots and lots of data and lots of replicates and different types of data. So I just stopped sharing my screen, but I'll reshare it and show one of the projects that kind of directly ties into these RNA modifications is one that we're taking advantage of a diversity panel of sorghum. So sorghum is this crop that's in the U.S. It's mainly used as a forage crop, but it's grown all over the world. And these pictures are from the deserts of Arizona. You can see it's pretty sparse out there. But in Arizona, the University of Arizona has an ag station in Maricopa, which is right outside of Phoenix. And at the Maricopa Agricultural Center, they have what they call the gantry. And this gantry is actually this huge phenotyping setup or platform, which can move up and down a field. It's a couple of acres, and typically they have sorghum planted in the field, but they put lettuce, they have done sunflower. And what this gantry does is, and you can see it here as well, it's about the size of a minivan. And what it does is it moves up and down the field and takes high resolution pictures and scans essentially of the plants that are growing. And you can actually monitor the growth of these plants, and you can monitor things that are happening kind of at the cellular level with these plants. And then we can tie this, hopefully, the goal is to tie this back to things that are occurring at the RNA level. So changes in RNA abundance, as well as changes in RNA modifications, and then see, like, essentially look for correlation between kind of, outputs of phenotyping, growth rates, like essentially yields, tolerance to a drought. So this field, they had half the field was drought stressed, half of it was well watered. The drought stress can be difficult to apply sometimes. Here I'm showing you the effects of a monsoon rain. It's where the field just got completely flooded. Luckily, this wasn't the year that that we were collecting the sorghum tissue. And here's Eric Lyons, who I learned genomics from, actually working out in the fields with Duke Pali, the two collaborators on the project. So to answer your question, we use a lot of phenotyping and kind of information about the plants and then we check and see what's happening at the cellular level. That is a hard word to say. At the cellular level, to see if we can correlate these two things. It's a lot of complicated math.
Yeah, this thing looks really neat. It seems like a giant version of what Magda is building, what you and Magda are using.
Yeah. So let me show you like just a few seconds of a video. So this is a courtesy of Duke Pali at University of Arizona. He's in charge of the Arizona portion of the Terra Ref project. And so you can see the minivan right there with a whole bunch of sensors on it. And you see, so this is a field of sorghum. This is obviously late in the season. And the gantry is moving left to right across the field. And then it's going to be moving up and down the field. And you can see that there's clearly some variation in how these plants are growing. Yeah, so it's quite an impressive facility. And so it's nice. Cool. It's nice, actually. I think it's important to be able to match what's happening in the field, where the reason you need so much information from the field is because there's so much going on. The environment has such a huge impact on what happens to those plants. And so you need what happens in the field, but you also need what happened in the greenhouse. You need the system that the Magda is developing so that you can compare between, it's almost experimental and control conditions. You have the field and the greenhouse, and then you're looking for, you're using those control conditions to help you narrow down to what's actually happening in the plants.
Yeah, very cool. And you're working on a few other crops too, right? I think cotton and
Wyoli. Yeah. So Wyoli. for time purposes, do you want to just do Wyoli?
Sure.
Okay.
Okay. We're about a half hour. So
yeah, exactly. Oh, let me get the right screen here. Okay.
And if we have time at the end, we could talk about cotton.
Exactly. Okay. So let's talk about Wyoli. So Wyoli is a really interesting plant. It's a desert shrub. It's native to northern Mexico and kind of southwestern U.S. If you were walking through the shrub, the fields of in this region, you wouldn't really notice it. It's not particularly showy. It's even the flowers don't really look like much, and so you would just kind of it's almost like oh gosh, tumbleweed. It's one of those types of plants. You just don't really notice it. It's what's interesting about Wyoli is that it produces an insane amount of rubber. It stocks up a lot of rubber in its stems. Typically in the winter, sorry, not in the winter. Typically, as temperatures start to shift and get cooler, it starts to load up on its rubber contents. And it's thought that this is a kind of a defense mechanism against the cold. And so what you can see here, so all of the gold labeled, it's not cells, but all the gold labeled particles are actually, those are rubber rubber particles. And so you can see that the rubber is kind of synthesized in the center area of the stem. This is a cross section of a stem, and then it's kind of transferred to the outer region, not quite in the bark, but towards that outer layer. And so some Wyoli can be, the stem can be between 6 or 8% rubber, which is pretty high for plants. And in addition, when the rubber has been extracted from these plants, you can actually use what's left over as kind of a natural biofuel. So so it kind of serves two purposes. So this is why JGI was really interested in working with us. So with Bridgestone, David Dierig at Bridgestone, Colleen McMahon at USDA, and myself, we spearheaded this proposal for JGI to actually sequence the genomes of these plants, because we don't really have a good genome. And if we want to figure out like what is actually working to turn on the rubber biosynthesis pathway in these plants, and figure out all the molecular components, we really need a genome. And it's been difficult. The reason that we don't have a genome is because our genome sequenced is that these plants have a really large genome. It's really complex. Their reproductive strategy kind of makes it difficult to get pure, basically simple genomes. So JGI, we're very happy that JGI is working with us because they have a lot of experience with complicated genomes. And so Wyoli is finally going to join the 21st century in terms of genomic resources. I should mention that Wyoli has actually been, it's been known as this alternative, this domestic alternative to for rubber production since like the 1940s. It was actually, there was interest in World War II, in creating a local rubber source as opposed to kind of sourcing rubber trees from Southeast Asia. Obviously, it was difficult at that time. And then, we've kind of gone through boom and bust in terms of using Wyoli as a rubber source. And so with Bridgestone's investment and with JGI's investments, hopefully this will make Wyoli a very tractable alternative to the rubber tree.
Yeah, so is the ultimate goal to try to up that percentage of rubber content?
Yeah, so what I know, and of course don't quote me on this, but I think we only need about 2% more rubber content in the stem for this to be economical. So Bridgestone is, I think, very confident that will happen, particularly through kind of advanced breeding efforts, using transformation facilities like Joyce's at BTI to take genes that we identify that are responsible or helping out with rubber biosynthesis and tweaking them so that they work better. So yeah, Bridgestone is really trying to turn this into a kind of an alternative crop for some of the crops that are already grown in Arizona. Because as we all know, the Colorado River is drying up. Cotton is going to become harder and harder to grow in Arizona. So we need a drought tolerant alternative for the local farming communities.
Right.
Okay.
Sense.
Yeah, it's a difficult task for farmers out in Arizona and West Texas to grow crops that require a lot of water, like cotton.
Okay, well, thank you, Andrew. That was a lot of neat stories in that half hour or so. Thank you so much. And I think now it's time to take some questions from the chat box. So if anybody has questions out there, feel free to throw them in the chat box. So there are a few in there now. One is non-coding RNAs are related in some way to epigenetics. Is that true?
Yeah, so that's absolutely true. Some long non-coding RNAs are responsible for or help facilitate the deposition of epigenetic marks. There's a really famous case of this in mammals. In females, like one of the X chromosomes is inactivated through a long non-coding RNA. So when you see, like, calico cats are a perfect example of this. I always use them when I'm talking about long non-coding RNAs and epigenetics. The reason that you have different colored fur is because different X chromosomes have been inactivated in each of those, like as the kitten, I guess, in the womb was developing. So absolutely.
Okay, so they can help silence genes or turn genes on, basically. What?
Yeah.
Epigenetics, that's kind of what we're talking about.
Yeah, exactly. Yeah.
And so are the RNAs modified by environmental cues, abiotic or biotic stressors? I think is that what you're trying to figure out? Or?
Yes, so we do know that some modifications. So there's some kind of specificity going on here that we don't quite understand, but there are subsets of RNAs that are specifically modified under different abiotic stresses. So for instance, heat, you get a very specific subset, in Arabidopsis, you get a very specific subset of RNAs that get modified that then go on to produce protein that helps deal with that stress, like heat shock proteins. The RNAs that encode for some of the heat shock proteins end up getting modified.
That's a good example. That's a good example. I think going back to the slide where you showed the potatoes and rice getting.
Oh yeah.
Do you know what was being increased to account for the higher weight? Was it protein, water, starch?
I am not clear. I think it was. Yeah, I don't want to butcher that explanation. I think it was just overall increase in cell density. But yeah, I don't want to butcher a very good body of work.
Drought stress in plants is related to the symbiotic relationships with fungi. Have you looked into fungal genetics?
That is a very good idea, and a very the endosymbiosis and the relationship between plants and kind of, I don't know if the correct term here would be rhizosphere, since it's everything that they're interacting with. I think that would be something really cool to study. It's super complex, and I think it would require some collaborations with some very intelligent people, because we're talking about determining which fungi are present, which bacteria are present, under which conditions, and then trying to track all that back to things that are happening at the RNA level. And I know that there are, yeah, it's sort of easy to do those in two-way comparisons, like what's happening specifically with the fungi and with the plants. But when you start getting these like these really complex networks, it just blows my mind trying to think about it. But I think that's where the future is, definitely.
Here's a neat question. Could you attach your lab camera setup to drones for assessment in fields in tree seedling plantations?
Oh yeah, so that's an excellent idea, except I think that they already have so these cameras are I don't know, like $5 maybe off of eBay, not eBay, but Amazon essentially. And these cameras, they're good cameras if you have a fixed location. I don't think they would be ideal for drones, but drone imagery or drone imaging has undergone, like, I mean, there's been a huge amount of improvements in the cameras that can, the weight and the resolution of the cameras that can be put on drones. And I think like, yeah, we should probably get a drone and just like have it hovering in the growth chamber. I think that'd be a cooler presentation, because those cameras are amazing. And on top of that, they can attach other types of sensors to those drones. Right. So yeah, it's drone imaging is very, very much state of the art. It's very cool.
It's part of digital ag. That's
yeah, absolutely.
These days. Here's a good one from Paul. Is the composition of the rubber from the different sources different, or is it just yield? So is it the same compound of rubber, or is it?
Yeah. Okay. Yeah. So each of these. Well, okay. Let's clarify. So if we're talking about a rubber tree, so the rubber particle is actually a mixture of different proteins and other molecules that come together to get you like this large, rubber goop. So it's protein and terpenoid type molecules and other things. And then the rubber that comes from Wyoli is slightly different. It's still obviously rubber, and you can still purify it to get the latex or the rubber that we would use. But it's slightly different. And the reason that I mention this is because of that the rubber from Wyoli doesn't have the whatever protein that causes latex allergies. So rubber from rubber tree has this protein that causes people to become very sensitive if they repeatedly wear, and not all people, but some people, to become allergic to the gloves they wear. Whereas rubber from Wyoli is naturally, it's missing this protein or compound. So I say that the rubber coming from each of the different Wyoli plants is the same, but rubber coming from different plants in general, like rubber tree. There's like dandelions that produce rubber. Wyoli produces rubber. There are a whole bunch of plants that produce rubber, and it's slightly different, but mostly the same.
How easily are the long non-coding RNAs identified, and how often are they associated with enhancer activities?
Yeah, so that depends on if you're talking about yeast, humans, or plants. So in mammals, link RNAs have been fairly well characterized or cataloged, I should say, and a lot of them, I don't know an exact number, but they are definitely associated with enhancer elements and enhancer-like activity. In plants, there's been a lot less. We know a lot less about this association between enhancer elements, so regions within the DNA that help to make a gene express more, and transcription coming from that specific region. So to call an RNA an enhancer RNA, I think we don't have those data yet. It's unclear, and I think some of the data from mammals is even maybe a little controversial as to whether it's actually the DNA element that's conferring this enhancing, or if it's the RNA coming from that element. But we don't really know in plants. In terms of identification, it's been kind of a long learning lesson for me, at least in plants. So I wrote got a proposal funded in 2018 where I said, we're going to identify link RNAs from the top 15 most deeply sequenced plants. And we're going to do it in 2 years. And we're just now like finishing up a project on identifying link RNAs in the mustard plants. So we've identified link RNAs in a bunch of different species, but it's not trivial. And of course, you want to be careful about what you've identified. You don't want to say that you want to make sure you're identifying long non-coding RNAs and not something else. So
yeah, it sounds like it's still early days.
Sort of, yeah.
In this field. Another interesting one, is there a gene known in plants homologous to the human RNA demethylase that you showed expressed in potatoes and rice, or do plants use something different?
So there are, the short answer is yes. There is so there are these 3 classes of proteins that are involved in RNA modifications. There are the writers, the ones that deposit the modifications. There are the readers. They kind of interpret those marks and they do different things with those RNAs based on the combination of marks. And then there are the erasers. And there are certainly enzymes that remove these marks, the M6A marks in plants and Arabidopsis. I'm not sure if it's exactly so I would say they are functionally homologous, but maybe not, like maybe they don't have a shared evolutionary past, but I might just be splitting hairs. But yes, there are these class of enzymes that will remove marks from these RNAs in a similar fashion.
Kind of related, what is your opinion or speculation about recent works that suggested the functional conservation of link RNAs despite the sequence divergences?
I feel like somebody's looking at one of my bioarchives papers. So I think that there are a number. Well, let's back up. So a link RNA is it doesn't need to maintain sequence conservation to produce a protein. So most mRNAs, you see signatures. Like if you look between close relatives, you can tell that these genes are related because they produce a protein, and so there are certain sequence constraints on those mRNAs. With long non-coding RNAs, typically they need to produce a structure, maybe, or they need to be transcribed from a similar location. So if they're regulating, the expression of a nearby gene, then all they need to do is be transcribed. If they're interacting with a protein, then they probably would need to conserve structure. And if they're interacting with, say, another RNA or a DNA molecule, then they would need to retain sequence or be conserved at the sequence level. So there's these different kind of gradations of conservation that's required for link RNAs to be functional. And in Arabidopsis and in the mustards, we found a whole bunch of long non-coding RNAs that were, say, transcribed and distantly related relatives. They're transcribed from the same location in the genome, adjacent to the same protein coding gene, but they themselves, those link RNA transcripts, didn't share any sequence similarity, suggesting. Obviously, we don't have functional evidence for this yet, but we're working on it, suggesting that these link RNAs may be what we called transcriptional syntalogs, meaning they come from the same location in the genome, they're transcribed, but they just don't share sequence similarity. And we think that these link RNAs are most likely going to be the ones that regulate adjacent genes, kind of regulate the transcription of adjacent genes. So, yeah, outside of my own work, there's some good work from mammalian link RNAs where they've shown that link RNAs with hardly any sequence similarity across all of the vertebrate lineage actually have conserved function. So they may have like this very small region of sequence similarity, but then when they go in and they do functional work on these link RNAs, they see that they are actually performing or interacting with the same genes or same proteins. So as usual, a lot of the molecular and functional work is coming from the mammalian fields, and we hope that a lot of it will translate.
Interesting. Okay, we're getting close to time, so I'll do one last question. There's still a lot of really great questions in the chat. Unfortunately, we can't get to them all. But this one seemed really interesting to me. Have you identified plant microproteins encoded by link RNAs in your work?
I really feel like somebody is reading. Yes, we have. And so this is actually work. Two different collaborative projects or work that was helped out by two different collaborations or two groups. So one, we essentially asked if any of our link RNAs in Arabidopsis overlapped with different sources of short peptides. So information about small peptides that could be mapped back to these link RNAs. And so Polly Sue, an assistant professor at Michigan State, she helped us identify essentially where in our link RNAs peptides might be coming from. So which of our link RNAs and where in those link RNAs they might actually be translated. So it now becomes a misnomer to say that these things are non-coding because some of them are actually producing very, very small proteins or maybe long peptides. And then Olar Skirich here at BTI with her postdoc from the Max Planck took a whole bunch of mass spec data and mapped that protein information back onto our link RNAs and asked if any of those, if any of the peptide data could map back. And so what we found was actually that I don't know, out of maybe 18 or 20,000 long encoding RNAs that we identified, probably about 200 had strong peptide data associated with them. And we then took an evolutionary approach. And we asked, like, are these peptides, are these open reading frames conserved? And is the peptide conserved, the amino acid sequence? And we found that for many of these, this short open reading frame, some of them, I think the shortest was 3 amino acids. Some were, I think a couple were in the 10, around 10 amino acids. So they don't have to be very long, but they were absolutely conserved. So it was really interesting kind of, side story in our identification efforts.
That's interesting. You might need a new term.
Yeah, I know. So there is a term that we're just going to
mostly
co-opt.
non-coding RNAs.
Yeah, mostly, yeah. So short open reading frames are what these are called, and it's yeah. We don't have a snazzy term yet. So we'll work on it.
All right. Okay. Well, that was great. Thank you again, Dr. Andrew Nelson, for taking the time to chat with us today, and thank you everyone out there for joining us. That was a really fun discussion. A lot of neat stuff and a lot of great questions. And please join us for BTI's next Breaking Ground discussion series, Wednesday, October 27, with BTI faculty member Sarah Evanja, who will be talking about GMOs. And that should be a good one. That's always a fun topic. And I'm going to paste a bunch of URLs into the chat box, and everyone. That come through? It did come through. Okay. And so that first one there is really long. That's to register for Sarah's Breaking Ground. It's going to be really good, so please don't miss it. And you can read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual-report. And the 2020 annual report is all digital with 3 different views. There's a single-page view, a 2-page view, which is most similar to like seeing a hard copy, like a double-sided PDF. And there's also a reading view, which is like reading an article on the web. And there are many links to videos and other neat interactive aspects in the online annual report, so please check it out. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. And thank you all again for joining us and for your interest and support of BTI. And thanks again to Andrew for being my guest here today. And yeah, have a wonderful day, everyone, and please be well. Take care.